Hey, it's Mr. Peterson. So just a short talk, quick run through of what we're going to do with this PTC lab. So this is the molecular structure of phenylthiocarbamide. Some of you are going to be able to taste it. Some of you won't be able to taste it. And from there we'll figure out who's homozygous dominant, who's heterozygous, and who's homozygous recessive. We'll do some little bit of some number crunching with regards to that. So anyway, the way the lab's going to work is like this. So let's back up. Let's just talk about your tongue for a second. You can pause the video and just read through this. And so this is, maybe you knew this, maybe you didn't, how your tongue works and how the senses of taste actually works, sending signals to your brain and where those taste receptors are. So just pause this, take a look at this. So here's the way the lab's going to look. You're going to extract some DNA. You're going to be the source of DNA. We're going to figure out what your genes are for this, for PTC, whether you have this gene for tasting or not. And we're going to extract some DNA from some cheek cells that you're going to scrape on the inside of your mouth. So take the blunt end of a toothpick, and eventually you're going to put that into PCR tube. PCR tubes are smaller than the microcentrifuge tubes that we've been using. They're thin-walled. They only hold 200 microliters. Coaching point, make sure that you're only writing on the sides because the top is going to be exposed to heat. So we'll use a Sharpie and make sure you have that label so you know which tube is yours. What we're looking at is chromosome 7. So chromosome 7 has the gene, so of the 23 pairs of chromosomes, chromosome 7 is where the location for this particular gene is. So if we zoom in on that gene, we're going to see a strand of DNA, and we're looking for the TAS2R38 locus, and at that locus there's a single nucleotide polymorphism. What does that mean? That just means that there's a single change in the gene. It either means you're a taster or not. A single change in nucleotide, an A and a T and a G and a C, and all those things. So if you have the taster, you have a C pairing with a G, a cytosine pairing with a guanine. And if you're a non-taster, you have a thymine pairing with an adenine at that location at the TAS2R38 locus. That's just a spot on chromosome 7. So that's what the genetics looks like. So what we're going to do is we're going to take this and we're going to do the PCR thing with this. You're going to take your DNA and we're going to amplify it with the PCR machine with a thermocycler, get a lot of it build up, and then we're going to do something called a restriction digest. And a restriction digest uses a very specific enzyme called an endonuclease. An endonuclease cuts, and the endonuclease we are using for this particular activity cuts specifically at that site of the TAS2R38 locus. So it's specifically designed to cut at that spot on the DNA. The thing of it is it only cuts the taster allele. So it'll break the taster allele, which is about 250 base pairs long. It'll break it into a 150 base pair and a 100 base pair chunk of DNA, but it won't be able to cut the T and an A. So the enzyme is specific to that locus, but it only cuts the C and the G. It only cuts that spot at that location. So it can't, the T and the A, the non-tasters, we're going to wind up with a bigger piece of DNA there, and that's how we're going to separate out your genetic codes. And eventually we'll take your PCR'd DNA and we'll do a gel electrophoresis. It'll be different than the protein electrophoresis. It'll be a horizontal gel, and we'll stain it with a protein. We'll stain it with a stain that will glow under UV light, and we'll be able to watch it in real time. It'll be pretty cool. So that, in a nutshell, that's what the PTC experimental plan will look like.